my group has been really interested over the last few years in the spectrum of congenital diarrheal disorders. These can have a really huge impact on the children whose fate is really blamed on a number of disorders. And this is a spectrum from very early onset inflammatory bowel disease to autoimmune enteropathies. And the ones that we focus on are direct defects in epithelial cells that cause problems with either barrier function or transporters. And this group has been expanding, and we've actually formed a consortium between UCLA, Vanderbilt, Boston Children's, and Toronto Sick Kids to take on specifically these epithelial defects. So these children generally are presenting in their first 1 to 2 weeks with overwhelming, often life-threatening diarrhea. And I'm going to focus my talk today on this disease, microvillus inclusion disease, or MVID, which is due to mutations in myosin 5B. So myosin 5B is a myosin motor, and a number of studies over the years have shown that myosin 5B can regulate epithelial polarity, specifically transport to the apical membrane in a number of epithelial systems. And then a few years ago, two groups, one in Germany and one working specifically on the Navajo Indian tribe, demonstrated that this disease, microvillus inclusion disease, was caused by mutations that either inactivate or cause knockout of myosin 5B. And indeed, in the Navajo tribe, this is about 1 in 10,000 births are homozygous single-point mutations in myosin 5B. The patients with this disease have a congenital secretory diarrhea that's life-threatening, it's unremitting, and really requires lifelong total parenteral nutrition or intestinal transplant, which is actually not available for the patients of the Navajo tribe, because if you're Navajo, you will not accept transplantation because it's putting someone else's spirit into your body. So this is a really high mortality, high morbidity disease. And the characteristic, which was described by Ernst Kutz almost 20 years ago, are these large microvillus inclusions that can be seen, although they really are only present in about 10% of the enterocytes. And it's really not been clear where these inclusions come from and how they relate to the disease. So several years back, we were successful in developing a myosin 5B germline and also intestinal targeted knockout mice, but I'm going to talk only about the germline mice today. They're born small, and they have a high level of stool water, which is how we define diarrhea. And so these mice are very sick, and they really die within 4 to 7 days of birth. The mice, as in the humans, show some very strong characteristics that characterize this disease. Here's a normal mouse intestinal epithelium. You can see these lovely microvilli. They're about a micron or more high. In the mice, you see that the microvilli are short. They're also a little bit fat. And you can see here a microvillus inclusion below the apical membrane. Here's a larger one over here. So the mice really have almost more of the disease, frankly, than the human children. And this is really shown here. Here's a normal intestinal mucosa with some apical markers, DPP4, an apical peptidase, the sodium hydrogen exchanger that's necessary for proper intestinal absorption of water. In the myosin 5B knockout mouse, I think you can see these dots here. These are all inclusions. And you can see that the DPP4 as well as the NHE3 are really not lining the apical membrane. They're subapical and internal. And this is shown in maybe a little clearer. Here's the normal mucosa where DPP4 and NHE3 co-localize at the brush border. You can see that the NHE3 here is kind of diffuse and subapical. There's a little bit in these inclusions that are really throughout the intestinal enterocyte. Now, those get stuck. Other apical transporters, such as aquaporin 7, another member of the brush border, it completely disappears, actually, in the myosin 5B knockout. So how does this all manifest? Well, you can imagine that two things are going on here. Here's a control mouse for aquaporin 7. And here's LAMP1, which is a marker of lysosomes. So you have a large fragmentation of the lysosomes that are probably eating up a lot of this aquaporin 7 that's not getting to the apical membrane. So I wouldn't be here if we didn't make enteroids, and we have done that using the classic methods that we've heard about already. Fortunately for us, the enteroids recapitulate a lot of the factors here. So here's normal enteroids from a littermate mouse, and here's the myo5B knockout mouse. You can see lots and lots of actin-positive inclusions throughout, and then in the mouse, and here's it is in the organoids. I think you can see these large inclusions, and you can see even some of these starting to form from the apical membrane. So these organoids have allowed us to do a number of things to try to evaluate where these inclusions come from. And I think you can see up here there's a nice omega coming off here and an omega coming off here. But there are also a lot of these big organoid inclusions that are away from the membrane. So we wanted to really look at how these inclusions are forming, because a lot of people thought they were just stalled insertion. So to look at these organoids in more detail, we looked first at the characteristics of what was getting to the membrane. So here's SGLT1, which is the glucose transporter that's required for water absorption. You can see it lines the apical membrane in the control organoids, but in the myo5B knockout mice, it's either internal or it's really lost from a lot of the organoids. This can be really I think you can see here pretty clearly that what SGLT1 is left is sort of subapical or diffuse. So like in the children, these transporters are not getting to the apical membrane. And if they don't get to the apical membrane, you can't absorb water. Now, the other characteristic that we found in the mice that was unusual in terms of transporters getting to the membrane was that here's CFTR and control enterocytes. Here it is in the myo5B knockout mice. And I think you can see here that although there is CFTR in these inclusions here, CFTR is very nicely still in the apical membrane. And so this disease is characterized by a lack of your ability to pull water in because you don't have the sodium and glucose transporters getting to the apical membrane. But CFTR, the chloride transporter, is still getting to the apical membrane, and that's responsible for water secretion. So these kids really have a double whammy of not being able to get water in. So we looked in the organoids from control and myo5B knockout mice. You can see CFTR in the control knockout mice, but you can also see CFTR in the apical membrane of the myo5B knockout mice. And then we were able to, we really wanted to visualize this a little easier, and this is one of those things you can do in mice but can't do in humans. We were able to cross the myo5B knockout onto the LifeAct EGFP mouse. LifeAct binds to F-actin, so you can actually visualize F-actin. So the straight LifeAct mouse has nice brush borders shown here with the F-actin. When you look at the LifeAct myo5B knockout mouse, you can see apical actin, but I think you can see there are lots and lots of F-actin positive inclusions throughout. So we could make organoids from these, and again, they maintain the F-actin at the apical membrane from the controls, but in the knockout mouse you get both a little bit of F-actin in the apical and lots of F-actin containing inclusions. So you can see this organoid swells when you add forskolin. This is activating CFTR to pump chloride and water. Same thing happens in the organoids, and this is all quantified. So the myo-5B knockout organoids maintain the CFTR at the apical membrane, just like what we saw in the mouse and also in the children parenthetically. Now, how are the inclusions forming? And so to really look at this, the only way we could do this convincingly was actually to go back in time to real tissue culture, and that is to put pieces of mucosa in Ussing chambers to maintain them and know that they're alive because of their short circuit current, and put 70 kilodalton dextran, so a large dextran, into the apical media. And in the myo-5B knockout mice, we were able to show that 70 kilodalton dextran was accumulated into these F-actin containing dextrans inside this F-actin containing inclusion. So this is actually a totally unexpected and unrecognized apical bulk endocytosis pathway. And the organoids have allowed us to visualize this a little better. So if you just focus here, you'll see these inclusions forming, and you'll see right about here an inclusion is going to form from a little omega and then punch in. This actually takes about 2.5 hours to happen. This is an extremely slow process, and it's so slow I added an extra movie bonus. Here, you'll see this one come off. Just maybe we'll do that one more time so people can follow the arrow. I hope you got that. That this really is a long extension and pinching off that happens. Really, it's over 2.5 hours, and these are big. So what you're looking at are inclusions forming that are really 2 to 5 microns in diameter. There's no trouble seeing that. So actually, this has allowed us to do some more mechanistic studies, and we wanted to look at the role of dynamin and another inhibitor of macropinocytosis, EIPA. And when you incubate these organoids in dingo, which blocks dynamin, you see this accumulation of these omegas near the apical membrane. But EIPA, which blocks macropinocytosis, which looks like it's a completely different pattern, was not blocked. So as I said, there's a consortium now, and I just want to end with 3 caveats. One, we can do all this stuff from human, and that's what we're doing. But I want to note that the level of differentiation is the problem for analyzing these children from their own organoids. And this is shown here is I'm not ragging on Intestinal. They've seen these things. So if you grow these things in Intestinal, you'd think this is a nice brush border. But if you then take Wnt out, you get a really nice brush border. So this is an immature brush border. And that's one thing that has to happen. The other thing is, even we've gone to these monolayer cultures, and they're great. But as you can see here, this is a great brush border by F-actin, but only one cell here really has a lot of apical DPP4 presented. So it's a real question of differentiation. And then lastly, culture can cure. So myo-5B knockout mice have a deficit specifically in tough cells. They don't have many tough cells. But if you grow their organoids in the standard media, as in just good old Intestinal, it cures the deficit of tough cells. So all this together says we've really got to be very careful in what we do. And as we go forward in trying to do high throughput screening methods and things like that, we have to know that we're at a level of differentiation that matches what we want to test if we're going to quote unquote fix things. And with that, I'll stop. Thank you.